The gram-positive cell wall structure includes the cell membrane, shown here in light blue, and the cell wall that's shown in dark blue. The gram-positive cell wall is very thick. It's made up of multiple layers of peptidoglycan. Peptidoglycan is composed of alternating molecules of NAG and NAM. The peptidoglycan layers are reinforced with teichoic acid and lipoteichoic acid, shown here in green. Here you can see another view of the gram-positive cell wall. The interior of the cell is represented in white. In the interior, you would have the genetic material and ribosomes and other things that the cell needs to function. The next layer out in light blue is the cell membrane, and on the very outside in dark blue is the cell wall. Gram-negative cell wall is different. This structure begins with an inner cell membrane, shown in blue, then a thin layer of peptidoglycan, the cell wall, and then a second membrane, the outer cell membrane. On the very outside is lipopolysaccharide, or LPS. Here we see a different view of the cell. The cytoplasm or interior of the cell is shown in white. Right outside the cytoplasm is the inner cell membrane, then the thin layer of peptidoglycan or the cell wall, then the outer cell membrane, followed by the very outside area shown in orange, which is where the LPS is sticking off of that outer membrane. Here we see a view where we have a side-by-side comparison of the gram-positive and gram-negative cell walls. You can see they both have a cell membrane. The gram-positive then has a very thick layer of the peptidoglycan or the cell wall material that's reinforced with the teichoic and lipoteichoic acids. On the right, you see the gram-negative cell wall structure, and it starts with an inner membrane, a thin layer of the peptidoglycan cell wall, then an outer membrane with LPS sticking off of the outside. Now let's have a look at how these two cell wall structures hold up in the gram stain. Cells are negatively charged on their exterior. The stains that are used in the gram stain are positively charged. They adhere very well to the outside of the cell. Specifically, they adhere well to peptidoglycan. The first step in the gram stain process is crystal violet. Crystal violet is the primary stain. When crystal violet is added, it's attracted to the exterior of the cells, and both cells will take on a purple color. This step is then followed by a rinse. In the second step of the gram stain, we add iodine, which is a mordant, and the iodine causes the crystal violet molecules to push deeper and farther into the peptidoglycan, specifically of the gram-positive cell wall. And that's because that peptidoglycan layer is exposed, and so it is able to push those molecules deep, deep, deep down inside that peptidoglycan. It doesn't have much of an effect on the gram-negative cell because there is no exposed peptidoglycan. So the stain remains the same. Both cells are stained purple at this point, and the iodine is then rinsed off. The next step is to add an alcohol. This is a decolorizer. Now, if your iodine step has been done properly, your color is not removed from the gram-positive because that mordant forces the crystal violet in deep enough that it doesn't come off. Contrariwise, with the gram-negative, that alcohol affects that outer cell membrane and it will dissolve it and any stains that are attached to it. And at the end of this step, the gram-positive cells are purple and the gram-negative cells are now clear. They are unstained because all of the stain has been dissolved away. In the final step, the counter stain, which is safranin, is added. Now the gram-positive already is holding, that cell wall is already holding as much stain as it possibly can hold, so it doesn't pick up much of that safranin. It doesn't change in color. But because now we have freshly exposed peptidoglycan or cell wall on the outside of the gram-negative, the counterstain or the safranin is able to absorb deeply into those cells and those cells will stain red. So at the end of the gram stain, you will have purple gram-positive cells and red gram-negative cells.